Hi, my name is Andrew Steen, and I'm going to talk about the idea that microbial enzymes in subsurface sediments have very long lifetimes because they're, structurally speaking, unusually stable. And this work was done with Taylor Royalty, Jenna Schmidt, and Karen Lloyd, all at the University of Tennessee. So I'm interested in looking at the properties of microbial extracellular enzymes because microbes in subsurface sediments or anywhere else often can't directly take up large molecules, which are often the most bioavailable molecules in an environment. And so they produce extracellular enzymes to break down these polymers into monomers or oligomers, which are small enough for them to take up and metabolize. So I really like looking at extracellular enzymes in especially subsurface sediments because they tell us a little bit about what the microbial community wants in terms of carbon and nutrients. So a few years back, a master's student in my lab named Jenna Schmidt measured activities of about a dozen different enzymes at site M59, which is in the Little Belt area of the Baltic Sea, just inside coastal waters in Denmark. And these were collected during IODP Expedition 347. What she found was that especially in the upper 50 meters of this sediment core, which the upper 50 meters were a little bit more organic matter rich than sediments below 50 meters. She was able to measure a wide array of extracellular enzymes, particularly peptidases. Those are the plots that are in green, to a lesser extent, glycosyl hydrolases, so enzymes that break down polysaccharides, and also extracellular alkaline phosphatase. So in these plots, the filled circles represent the live samples and the open circles represent our killed control. The way we produced the killed control was we autoclaved sediments for an hour on a liquid cycle. So these sediments had been cooked at 121 Celsius in the absence of oxygen for an hour. And what we noticed is that it looks a little bit like the killed control, despite that pretty harsh treatment, kind of matches the shape of the live sample. So you can see here with gingipane, for instance, when we get a bump in the live sample, we also get a little bit of bump in the killed control. You can particularly see this with ornithyl aminopeptidase. And you can see this more clearly in a plot of the activity of each live sample versus the corresponding autoclave sample. And what we found is a loose but statistically significant relationship between activities in live samples and their autoclaved counterpart that spans like 4 orders of magnitude of activities. We plot these data in yet a different way, just as the percentage of activity remaining after autoclaving. And you can see that the numbers range pretty widely, and we have a few data points that are up above 100%, which is probably an artifact of noise associated with low activities, low initial live activities. But for very many of these samples, some substantial fraction of extracellular activity is retained upon autoclaving. The median value is something like 25%. And that suggests an incredibly stable enzyme. So, you think about, well, I should say that we expect a really stable enzyme because we know that the carbon turnover times of the microbes that are in these sediments are quite long. So this is a plot taken from Matra et al. 2019. And I want you to pay attention to the blue filled circles right here, which represent the turnover times of live biomass. And you can see that a lot of these turnover times are longer than a decade. And so if you have a microbe that's turning over its organic carbon on a time scale of a decade, it's got to have enzymes that are active in order to stay alive. So this microbe itself must be producing enzymes that are also active on a comparable time scale. We wanted to get an estimate of what the turnover times of the enzymes in these Baltic Sea sediments that we measured are. And so we took a sort of a box model approach. This is just like a standard kind of reservoir residence time calculation, where we say that if the extracellular enzyme activities are in steady state, that is to say that if the enzymes are being degraded at the same rate that they're being produced, then we can define a turnover time tau, which is just the concentration of enzymes divided by the enzyme production rate. So this is a nice model. I was happy to sort of conceptualize the problem in this way. The only problem is that we cannot measure the enzyme concentration directly, and we also cannot directly measure the enzyme production rate. So to get a handle on this, we decided to make some sort of limiting assumption. So we say, imagine that 100% of the microbial community biomass production goes to producing extracellular enzymes. Now, obviously that's an overestimate, but it causes us to underestimate the actual turnover times. And so in that sense, it's a conservative approximation. So then we can measure tau as enzyme concentration divided by the biomass production rate. Again, we can't measure the biomass production rate, but we can model the respiration rate of the community and then set biomass production rate as being equal to the community growth efficiency times the modeled respiration rate. Now, coming back to the enzyme concentration, again, we can't easily measure that directly. But we can measure Vmax, or the maximum potential activity of enzymes. And then we can define a specific activity, which is the essentially the activity per enzyme. And so divide Vmax by specific activity, and we get an enzyme concentration. Now, again, we can't directly measure the specific activity of the enzymes in this environment. But a lot of times when people purify and characterize a new enzyme, they will report a specific activity for that enzyme. And that specific activity is likely to be larger than the actual specific activity in the environment due to things like inhibition of enzymes by humic substances in the environment, and so forth. So again, this overestimate gives us an underestimate of the enzyme lifetime. So again, it's using these literature values as a conservative thing to do. So we can take these specific activities that are reported when purified enzymes are characterized, and we look at similar enzymes to what we've measured. We can get a growth efficiency estimate from the literature, and here we relied on growth efficiency measurements in the Sargasso Sea, which is an area a little bit similar to subsurface sediments where growth rates are very low. And then we can model respiration rates using geochemical profiles. So we put all of these things together and we get a what I think it's fair to call a back of the envelope calculation, but it's a conservative calculation of the enzyme lifetime, which this is the overall equation for that. And depending on the value of specific activity for clostrapane, which is the enzyme that we chose to look at, we get turnover times on the order of months or years. The median value that we see here is 7.5 months. And that's a really long turnover time. So if you think about if you're doing some kind of molecular biology work in the lab and you accidentally leave your enzymes on the bench overnight, they're probably shot in the morning, right? A lot of times we think about enzymes having lifetimes on the order of hours, not months or years. But it would be nice to get an actual measurement of these enzyme lifetimes. So the direct approach to that would be to heterologously express enzymes that we find gene sequences for in subsurface metagenomes and actually measure whether enzyme lifetimes in subsurface sediments are longer than anywhere else, for instance, surface sediments. And we went down this little path a little bit with University of Tennessee graduate student Eric Zhou, who's quite good at heterologously expressing proteins. But that's really tough to do, or at least it can be tough to do cleanly, as shown by this gel, in which we can see that we had This top band is the protein that we're looking for, and these green and red bands are a bunch of other crap that Eric and postdoc Taylor Royalty were struggling to purify. And of course, we can put in a lot of work and get one enzyme, and then we'd have to put in a lot more work to get another enzyme and so forth. So we went looking for a faster solution, and we identified a computational workflow that can give us an estimate of enzyme stability. So we start with a protein sequence or an amino acid sequence that we get from a metagenome. And then we feed that sequence to the newly released AlphaFold software. This came out this spring, that uses deep learning to create what structural biologists think tend to be pretty accurate predictions of the structures of previously unknown enzymes. And so we get this really nice hypothetical protein structure from AlphaFold. We take that structure and feed it to a software like FoldX, which looks at all of the energies of interactions among residues within the protein and between amino acid residues and water molecules, and comes up with an estimate of all of the energies of all of these interactions. And from that, the software can calculate a free energy of folding of the enzyme. That is to say, how much free energy is released when the enzyme goes from the unfolded to the folded state. And that is a measure of the thermodynamic stability of the enzymes. So if we do this for a whole bunch of proteins in deep sediments and surface sediments, we should be able to test the hypothesis, at least computationally, that subsurface enzymes are more stable. We looked at, again, genes taken from the site M59 in Little Belt in the Baltic Sea, and we looked at two of the most abundant secreted proteins. In this case, they're two glycosyl hydrolases. And we find that indeed for the most abundant class, GH103, which is a peptidoglycan-lytic transglycosylase, indeed the free energies of foldings are lower in deep sediments than in surface sediments, implying a more stable protein. And when we look at pairs of homologs in those sediments, so the blue and red lines here connect homologous proteins between the deep and the surface sediments. You can see that there's a lot more blue lines indicating a more negative free energy of folding in the deep sediment than the red line. So this, and this turns out to be true also for GH29, the second most abundant secreted hydrolase. In this case, it's an alpha fucosidase. That it seems like these proteins are more thermodynamically stable in deep sediments than in surface sediments, which is an exciting finding because it suggests that protein stability is an important trait that's subject to selective pressure as these communities evolve from surface sediments to deep sediments. So going forward, that raises a bunch of interesting questions. We measured stability of subsurface enzymes at one site for two classes of enzymes. We're looking at more classes of enzymes at that site, and we've politely asked the National Science Foundation for money to look in some other sites. It'd be nice to know how widespread enhanced stability is. It'd be nice to know what the physicochemical mechanisms account for that stability. And then what are the limits of intrinsic protein stability, and what other factors other than protein structure can account for that stability? So for instance, with extracellular enzymes, we know that sorption to mineral surfaces increases stability. Within the cell, we know that for cytoplasmic enzymes, trehalose and other osmolytes contribute to protein stability. And it would be nice to get some quantitative evidence of how important each of these factors is. So put these together, and this can tell us a little bit more about what the important biochemical adaptations are to slow life in the subsurface. So with that, I'd like to thank funding, which comes from the DOE, from the National Science Foundation, and CDEBI, and the Deep Carbon Observatory, and within the University of Tennessee, the Joint Institute for Computational Science, and a bunch of great collaborators I've had inside my lab and outside of my lab that have helped with all of this work. Thank you very much.